Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Human Astrovirus: Structure, Transmission, Pathogenesis, and Prevention. Table of Content. Introduction. Structure of Human Astrovirus. Genome of Human Astrovirus. Transmission and Epidemiology. Replication. Pathogenesis. Clinical manifestation. Diagnosis. Treatment. Prevention and control. Recent development. Introduction. Human astrovirus is a significant viral pathogen belonging to the Astroviridae family and the Astrovirus genus. It is known to cause gastroenteritis, particularly in young children, the elderly, and immunocompromised individuals. The virus is highly stable in the environment and primarily transmitted through the fecal-oral route, contaminated food, and water. Structure of human astrovirus. Human astroviruses are small, non-enveloped, spherical viruses measuring approximately 28 to 35 nanometer in diameter. They exhibit an icosahedral symmetry and possess a single-stranded RNA genome with positive polarity. The viral particles have a characteristic 5- or 6-pointed star-like surface pattern, which inspired the name Astrovirus, Greek, astron, star. The surface appears rough, with small protruding spikes, and the virus has a buoyant density ranging between 1.35 and 1.37 gram per milliliter in cesium chloride. Capsid proteins and maturation. The capsid precursor protein, consisting of 180 copies per viral particle, undergoes cleavage by host caspases during maturation. Infectious particles are produced by further proteolytic processing of the VP70 precursor protein by extracellular proteases, resulting in 3 structural proteins. VP34 forms the capsid shell and is derived from the conserved N-terminal region. VP27 and VP25 derived from the C-terminal domain, contributing to spike formation. There are at least 8 distinct serotypes of human astroviruses, human astroviruses 1 to human astroviruses 8, distinguished by genetic and antigenic variations. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Genome of human astrovirus. The HASTV genome is a linear monopartite single-stranded RNA plus molecule, ranging between 6.8 and 7 kilobytes in length. The 5' terminus is linked to a viral protein genome-linked VPG protein, while the 3' terminus features a poly-A tail. The genome serves both as a genetic template and messenger RNA for viral protein synthesis. Open reading frames, ORFs. The genome consists of 3 overlapping open reading frames. ORF1A encodes non-structural proteins, including viral proteases. ORF1B encodes the RNA-dependent RNA polymerase, translated via ribosomal frameshifting. ORF2 encodes the VP90 capsid precursor protein, expressed from a subgenomic RNA. Transmission and Epidemiology. Human astrovirus spreads predominantly through the fecal-oral route, person-to-person contact, and ingestion of contaminated food or water. Studies indicate the virus is highly stable in various environmental reservoirs, including drinking water, fresh water, and seawater. The virus exhibits a global distribution and disproportionately affects young children, 1 to 3 years old, though outbreaks have been reported in adults, particularly in Japan. Seasonal and geographic patterns. Human astrovirus infections peak in winter in temperate regions, similar to rotavirus. The virus is responsible for 4 to 8.6% of diarrheal cases in children, with variations based on geography. It has been linked to foodborne gastroenteritis outbreaks in adults and school children. Immunocompromised individuals, such as AIDS patients and organ transplant recipients, are at a higher risk of severe infection. Replication cycle. 1. Attachment and entry. Human astrovirus binds to host cell receptors and enters via endocytosis. Second, uncoating. The viral RNA is released into the cytoplasm. Third, translation and polyprotein processing. The genome is translated into 2 large polyproteins, non-structural protein 1A and non-structural protein 1A or 1B. The viral proteus and host cellular proteases process these polyproteins into functional, non-structural proteins. Fourth, genome replication. A full-length negative-sense RNA template is synthesized. This template is used to generate new positive-sense genomic and subgenomic RNAs. Fifth, capsid assembly and maturation. The capsid precursor, VP90, assembles into immature virions. VP90 is cleaved into VP70 by caspases. Trypsin further processes VP70 to enhance infectivity. Sixth, release. The virus exits the host cell via lysis or non-lytic mechanisms. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Pathogenesis. Human astrovirus primarily targets intestinal epithelial cells, leading to destruction of enterocytes, disrupting absorption and secretion functions, inhibition of epithelial permeability, contributing to diarrhea, systemic spread in immunocompromised individuals, though rare, both humoral, IgA, and cellular, CD4 plus/CD8 plus T cells. Immune responses play a role in protection. The virus is excreted in high concentrations, up to 10 to the power of 13 genome copies per gram, and can persist in stool for up to 2 weeks post-symptoms. Clinical manifestations. Incubation period, 4 to 5 days. Symptoms, watery diarrhea, lasting 1 to 4 plus days, abdominal pain, fever, anorexia, and occasional vomiting. Severity, milder than rotavirus, severe cases in malnourished children and immunocompromised patients. Shedding duration, 1 to 2 days pre-symptoms, 4 to 5 days post-recovery. Diagnosis. Electron microscopy, direct detection in stool samples. Enzyme immunoassay, uses monoclonal antibodies for virus detection. Latex agglutination test, specific for HAS-TV-1. ELISA, and immunofluorescence, detects HAS-TV antibodies. Reverse transcription, PCR, gold standard for genotyping and detection. Treatment. There is no antiviral treatment available. Management includes oral or intravenous rehydration therapy to prevent dehydration, supportive care to manage symptoms, prevention and control. Currently, no vaccines exist against human astrovirus B. Preventive measures focus on interrupting transmission routes, hygiene practices, proper hand washing and sanitation, food and water safety, routine testing and decontamination, disinfection, use of appropriate disinfectants for contaminated surfaces. Recent developments. Studies on human astrovirus variants, emerging strains with altered pathogenicity and zoonotic potential are under investigation. Vaccine research. Efforts are ongoing to develop an effective vaccine focusing on capsid proteins. Environmental surveillance. Detection of human astrovirus in wastewater, suggests potential use as a marker for viral gastroenteritis outbreaks. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!